Hi, so my name is Jenny Ding, and I'm going to be doing my lecture project on infectious diseases, which we went over in Bio 172 recently. So, infectious diseases can be caused by three things that we went over: eukaryotes, bacteria, and viruses. So, over here is the Plasmodium parasite that actually is a eukaryote that can cause malaria. And this picture right next to it over here is the bacteria that causes tuberculosis. This last one down here is the HIV virus that causes AIDS. So first I'm going to talk about the eukaryotic infection, and my example would be malaria. So malaria is caused by Plasmodium, as I said, and it is transmitted through mosquitoes who are Plasmodium infected. And these mosquitoes will inject the Plasmodium parasites into the humans as they feed on that blood. And the Plasmodium parasites then target the liver because it is rich in red blood cells, which is what they attack. And this causes kind of a cycle because the mosquitoes who feed from the infected human will also be infected with the Plasmodium, and they can continue to transmit it. This Plasmodium is a protozoan, which is a single-celled eukaryote. A eukaryote is an organism that has a nucleus that is membrane-bound. And this is different from bacteria, which is a prokaryote. So, talking about tuberculosis, it is caused by a bacteria, and tuberculosis remains asymptomatic, meaning that there are no symptoms in many cases, and people who have tuberculosis might not even know it. And it becomes active when these people have compromised immune systems, which allows tuberculosis to kind of overcome those defense mechanisms as they are weakened. And when it overcomes those defense mechanisms is when it finally shows up and starts to show symptoms that it has. So next is the viral infection that I'm going to be talking about, and for this example I'll be using HIV. I'm going to put a lot of emphasis on this one because we actually went over this more than bacteria and eukaryotes in lecture. So first I'm going to introduce what a virus is. So viruses have genetic material, and this can be either DNA or RNA, and the DNA or RNA can be double-stranded or single-stranded. They are super tiny and ubiquitous. Ubiquitous means that they are everywhere and they can infect any live organisms, including animals, plants, fungi, bacteria. They are pathogenic, so they cause diseases. And by infection, they can influence ecological and evolutionary processes. They are obligate parasites, which means that they cannot complete their life cycle without exploiting a suitable host. So they need a live organism to actually complete their life cycle. And because of that, they are not alive, and they are non-cellular. They cannot produce their own energy, and they're not capable of transcribing or translating, which is the process of making proteins and all the things that they would need to be producing if they were alive. And for that reason, they are not given scientific names. So here I have some examples of viruses, and I'm going to specifically be talking about this one here on the left, which is the HIV virus, the human immunodeficiency virus. But first I also want to talk about some virus pathways. So viruses have the opportunity to go lytic, but they may not choose not to, and that means that they can go over here and have the lytic pathway, or over here have the lysogenic pathway, or they can even have both. But all viruses at one point in their life cycle will have the opportunity to go lytic. So the lytic pathway is the one where the viral genome is replicated using the, in this case, it is a bacteria that it's infecting, and the virus is up here. But it will use the bacteria to replicate and synthesize its proteins, and it can then assemble these proteins and everything else, all these parts it can assemble to produce more viruses, and the cell that it's in can break open, and all these mature viruses will be released and can attack other cells, and it can continue happening until, it causes a serious problem, like for example in a human, where it can cause a disease. And then it can also go and take the lysogenic pathway, which is where the viral DNA is actually integrated into the host cell chromosome. So its DNA is kind of blended into the host DNA, and this will go undetected. The viral DNA is then, every time the host DNA is replicated, then the viral DNA will also be replicated, and then it can be reproduced through that way. So my example for the viruses is HIV, and HIV is a retrovirus. Retroviruses have this special enzyme called reverse transcriptase, and what reverse transcriptase does is it can take RNA and turn it into something that's called cDNA, which stands for complementary DNA. cDNA is just DNA that only contains coding sequences, so coding sequences code for things such as proteins, transcription factors, and it will only have those coding sequences. So that means that HIV's genome is actually made of single-stranded RNA, and when the reverse transcriptase produces the cDNA, it can go lysogenic, and as we said here, it can enter the host cell chromosome and just kind of blend in. If it was RNA, it would obviously be detected because that's completely different from DNA, and so by having the cDNA, it blends in and can go undetected. So HIV can lead to AIDS. HIV stands for human immunodeficiency virus, and notice that this is not a scientific name, so it shows how the virus is not alive, and it can be transmitted through the exchange of bodily fluids, which makes HIV an STI. So HIV actually attacks T cells, and T cells are crucial for recognizing pathogens and producing immune responses, which means that HIV infection can lead to AIDS, which stands for acquired immunodeficiency syndrome. And this just means that it interferes with the body's ability to fight off infections and can leave an individual really weak. And by this infection, it can kill cells indirectly. So it's not the HIV virus that actually kills the cells, but the immune response that kills HIV-infected cells that actually ends up leading to the death of those cells. So one thing I wanted to address is that antibiotics cannot treat viruses. So antibiotics actually target bacterial enzymes and metabolic pathways that are specific to the bacteria. So taking antibiotics inappropriately can actually lead to antibiotic resistance, which is a huge problem, and which is actually an increasingly predominant problem. But luckily, we do have vaccines, and vaccines are injections of attenuated, which are weakened, which means the same thing as weakened, or dead virus or bacteria. So whether it's weakened or dead, it exposes the body to that pathogen and strengthens the immune response of the body. So the body essentially gets to practice recognizing and fighting off that pathogen. So when it does come face to face with the full-blown pathogen, it has already built a stronger immune response as to where it would have a weaker one if it was the first time that the body had encountered that pathogen. And this is why yearly vaccinations are needed, because pathogens can actually mutate, and to keep up with those mutations, we get yearly vaccinations, such as the influenza vaccines. If you get yearly influenza vaccines, the reason why is because we need to catch up to those mutations. And some pathogens can actually mutate really rapidly. For example, reverse transcriptase is extremely mutagenic. It mutates very quickly and very often. So that is why it's been difficult to produce a vaccine for HIV. Okay, so that is the end of my lecture, and I just wanted to say thank you to you, Dr. Carpenter, for your efforts, because I'm really glad that someone cares enough to try to make a class for students in order to help them be more successful. And I wanted to say good luck to you for your future endeavors in this class and beyond, and I hope you have a wonderful break.